Hello, this is Dr. Michael Edwards, founder and CEO of BioInfo Solutions, a data analytics company based out of Parker, Colorado. This entry is part 2 in a series of instructional videos detailing a meta-analysis on 8 different human gene expression studies looking at papillary renal cell carcinoma. In this installment, I give a demonstration on collecting gene expression data from a single disease using Illumina's BaseSpace Correlation Engine. I also describe the types of biological information we can obtain from this type of meta-analysis, as well as go over exporting the results to other statistical and visual programs for further work. A link to the actual meta-analysis used in this video can be found in the description of this YouTube video, although you do need an existing correlation engine account. This is the BaseSpace Correlation Engine platform homepage. Given up here are a few categories you can use to search their extensive database. But what I want to point out in particular is this number here, 21,995. This is the amount of studies in this database that we can actually search through. So what we're going to do for our meta-analysis is we are going to basically look through these approximately 22,000 different omics studies within their database and try to pull out anything related to papillary renal cell carcinoma. So in order to do that, what we do is go up to curated studies, this folder looking thing here, and click on that. Now that I'm at this window, what I can do is basically search their database and pull out studies that relate to what I'm looking at. So in this case, in this query window, we're interested in kidney cancer. So I'm going to start kidney, and I type cancer. And you'll notice as I type, these terms start coming up that are related to what I'm typing. So again, this is a curated database. So it's already been populated with biological terms associated with those particular studies. So when I click on kidney cancer, all those studies associated with this particular phenotype are going to show up. And now we've gone from approximately 22,000 studies down to 138. 138 is still a lot to go through, so I want to filter this even farther. So I'm going to go down here under curated studies. I'm going to hit organisms. I'm only interested in humans, so out of these 138, there are 131 that are based on human studies. I can go to data types here. For this particular meta-analysis, I'm only concerned in RNA expression data. So what I'm going to do is click on that, but you can see you can look at mutation data, microRNA, there's methylation. Now, for keyword, what I want to do is put in a term here that's going to filter this more to exactly what I want. Again, the rare cancer we are concerned with is papillary renal cell carcinoma. So I'm going to put in papillary. And as I type that, terms come up. I'm not going to select any of those because there's more than one papillary cell in different parts of the body. I'm just going to hit apply filter. All right. As I do that, now we have 11 studies we have to go through. These are the studies that we're going to use for our meta-analysis. So now what we have to do is kind of go through there and basically pick out the comparisons that we feel best represent what we're looking at. And we're going to use all these different comparisons to combine into our meta-analysis. We can take a closer look at this first entry here. This is a study from the Cancer Genome Atlas, or TCGA from short. Again, this is an RNA expression study. If we want to get more details on this study here, all we have to do is click this button here, Full Study Details, and it should kick you out to this window. On this tab, it's giving you more information, more in-depth information on this particular data set. As you can see, what's nice about this tab is a lot of times it'll give you links to external pieces of data. In this case, this is a wiki page describing the TCGA data set. On some of the other studies, what you'll find is it'll give you a link to where they actually deposited the raw data in some public repository, something like Geo. There will also sometimes be links to the actual manuscripts where this data was used in. So that can be nice, seeing what their interpretation was compared to yours. But for our meta-analysis, what we're really concerned with is this tab here, the Biosets. And let me click on that. So the nice thing about Correlation Engine is they've already done the statistics for you. Again, this is a curated data set. So what they do is go in and look at all the variables associated with a particular study. From those variables, such as disease versus control, they will already do the comparisons for you, basically doing t-tests for you. Given in this TCGA data set are all these different types of cancer that they looked at compared to the normal version of that tissue. So in this example here, we can look at bladder cancer tumors versus normal bladder tissue. In this comparison, there were 223 samples used that are associated with both phenotypes. From this comparison, they found 9,306 genes that were differentially expressed. The criteria for a gene to be differentially expressed in one of these biosets is that it has to be, the p-value has to be less than 0.05, and the fold change has to be greater than 1.2 in either direction. You can click on any of these biosets to look at what kind of genes are in there. And you can download any of these data sets. But for our purposes, what we want to do is go back. So these are the data sets that we are going to pick from all these different studies. And what we're going to do is select only those comparisons that are relevant to what we're studying. In this case, kidney, renal, papillary cell carcinomas. So let's go back to our original curated studies and start picking out genes for our meta-analysis.